what are the consequences of saturation and underexposure in our images where we have no usable information? So let's have a look at that. What does that mean? So the consequences of saturation. In this image, we're using a spectral detector, and this spectral detector we'll see later today. I think, yes, today. We will see later what that means. A spectral detector, some of them have up to 34 channels, but in this case we have 32 channels. So we have 32 images, each of them representing 10 nanometers of the spectrum, 32 images of the same bead. So we have only one bead, okay? And we have 32 images in different colors of the same bead. But something is a bit bizarre, because in some images the bead has a certain diameter, in other images the bead looks bigger. So there's a different diameter in the different channels. How can that be? We have only one bead, so they all should have the same size. Well, if we apply the saturation and underexposure lookup table, we can see that some of the channels are saturated, and this is the reason. So these saturated objects will appear artificially larger. Okay? This means that not only we get the wrong intensity, we also get the wrong size. And then if the objects are close to each other, they will start fusing like this. Therefore, we also get the wrong number of objects. So it depends, because those that are far away from each other, we're going to count them properly. If we just count and we don't look at the intensities are definitely wrong. But if they're close to each other, then you're going to get the wrong number of objects. So keep the saturation and underexposure on all the time, LUT on, and then set the parameters to have no saturation in the area of interest. And now, what are the consequences of underexposing? So here we have a strongly underexposed background, so it's like solid blue. And this is often the case. I mean, the background is the dimmest area of the sample, so therefore this is the background that is underexposed. And why do we care about the background? We just want it to be black, right? Well, the problem is that when we're imaging a bead, I don't know if you remember, but I said the edges are in microscopy are never sharp. We never go from one pixel is black and the other one were inside the nucleus. It doesn't work that way. The edges are the contrast is not so well preserved, and so the edges are always a little bit a little bit blurry. And so where does this start and end? If we want to figure out the size of the bead, we have to quite precisely measure, decide where the start of the bead ends or not. But if we have a saturated, an underexposed background, then the edge looks retracted like this. And why is that? It's because all these very dim pixels here that are around the bead, we are basically telling the detector, do not detect them. So it doesn't. So we are setting our parameters in such a way that the detector is unable to detect the dim details in our image. So that's not very good. This is not detected, so you cannot do anything afterwards. So there is no details at the edges, and we get the wrong size. So we are going to have smaller objects instead of larger with saturation. So when we want to publish, we're sending a picture like this. The manufacturers, they ask you for your original images for the pictures that you put in your paper, and they have Fiji. They have very good software, so they will definitely know the high-low filter, and they will turn it on, and they will see that, and will go like, If you're claiming anything about size, this should be rejected. Okay, so it's really important that you do not do that. This one has a little bit of blue in the background. I don't know if you can see it. This one would certainly not attract any comment. So tiny bit of blue away from the object of interest is still acceptable. There's no reason to do this. So you better just have as a rule, don't. And so keep the saturation and auto exposure LUT on all the time. Yes, Tosca.
Yeah, I was just wondering, based on also the lecture yesterday with background, maybe I'm understanding it wrong, but I thought you want the intensity value in the background to be as low as possible for like a reduced signal to background or for your signal to background ratio?
So not zero.
Okay,
We want it to be as low as possible above zero.
okay, okay,
We want to remove, so we want to increase the contrast. So we want to remove all the possible sources of things that produce fluorescence and that is not what we want, basically. But we do not want any zero.
okay.
Yeah, because if you have zero, we have no, sorry about that. Sure. Ah, it happens every day at the same time. It must be everyone who's calling me. Okay, so if we have no details at all around the objects if it's like this. So I will show you another slide where you will understand what I mean. But so no zero. In a camera-based system, we will see why, but in a camera-based system, this is not possible. You will always have some sort of levels, but on a single point confocal, you can underexpose using the offsets. So set the imaging parameters to have no underexposure, and then later during the day, we'll go through the workflow of which parameter does what, and when you want to set it where. So to sort of anchor everything, and hopefully you will, it will crystallize things. So now the thing is with the background that you have left in your image, doesn't matter where it comes from, is that you can easily remove it after acquisition, and that is valid. So but the thing is that once you have this image, and you see in here, there's a high autofluorescence of the mounting medium, for example. So the background is gray. It's not a very pleasing image in the eye, but it doesn't matter because we can remove the background, we can subtract the background, and clearly we keep this original image. What is the difference between acquiring an image like this, subtracting the background, or acquiring directly like this? It's that in here you have all the details, all the fine details. When I am subtracting the average in this area, I'm going to preserve all the fine detail at the edges, but I will make the background darker. So that is the main difference, and it's a very important one, because in here you're not preventing the detector from acquiring the dim signal in the image. You don't want to do that. So we keep the saturation lookup tables on. We use the correct exposure. There's no red and there's no blue. So right now in this image, the saturation and underexposure lookup tables are on, but you don't see any blue and any red because it's correctly exposed. And then afterwards, we're going to subtract. So to acquire images without underexposure on single point confocal, this is where I said there is a risk with the underexposure. We have to look at the offset. Okay, the underexposure is not possible with cameras, and that's because camera manufacturers do not want any zero in the background because the electronic cannot deal with it. So they add a certain level of add an offset. So even if you acquire an image on the camera system that is without light, you will see that the background is around 100 count. Okay, so with a camera, not possible to be underexposed, but a point confocal, it's easy. Always subtract the background, so which includes, like this, would include the camera offset and the mounting medium. Autofluorescence, for example, you subtract it after acquisition. And then you treat samples and controls exactly in the same way, and you need to say it in the material method of your paper that you have done this manipulation. It's very important. So the intensity, the consequences of saturation, and yes, Nadia? Did you want to say something, Sadia?
But I was saying that unless it's like a material method paper, I have never seen anyone write about these things. They write it like very vaguely, just write the instrument name.
Yes. No,
And
yeah, you're pushing it.
yes,
If they just write the instrument name, it's a very poor paper.
yes, and then usually like the papers have like a word limit. Every
Yes,
journal has a
absolutely.
so
Yeah.
this is why they try to take away as much information
Yes,
as possible.
absolutely, absolutely. but there is a major and documented problem with reproducibility of science. So there are several articles that are actually studying articles and looking at the material method and reporting that there is a major issue with reproducibility because the information that we have in the articles is not sufficient. However, if you look at these papers, which are very interesting, it's very interesting to read it, you will see that it's far from the majority. It's a lot of papers do not report properly, but it's far from the majority. It's about depending on what parameter you consider, and this was papers for light microscopy specifically, but there are other papers in other areas. Let's say it's about 33%, I think, that are missing some information. So that means that most people do their job. Okay, so you may not notice this type of information because sometimes they can be under the like in the figure legend that you would say these images have been, the brightness and contrast has been adjusted. It's just one little information, one little sentence like this, and it may be embedded within the paper itself without being in the material method.
I mean, there was one time in one of my papers, they commented that, okay, you need to give like nicer picture for the publication. I mean, you have your data set. So then they told us to like make the images prettier. It was the simple thing. So then I remember like the postdoc that time in our lab said in our lab, he sort of like adjusted the brightness and saturation and make it look pretty, and that was published. But then the data set for analysis was still fine.
Yeah, no, absolutely, absolutely. So what you're analyzing, and we will talk about this next week, but when you acquire, even for acquisition, when you acquire an image for analysis, and you acquire an image to actually put the image in the paper, you don't set things in the same way because your goal is not the same. So it's very important when you acquire an image to think about what the goal is. You cannot acquire all your data as if you were going to publish all these images because mostly your experiment and your time require that you go faster, for example, or you cannot put so much light on your sample. So you have some data sets to analyze, and you're going to have a few images that are meant to be published in the article. But when you publish it in the article, you cannot just publish. So sometimes you see that. You see an article where you can hardly see what they have in the images. And clearly something is wrong with the way they have displayed the images, and that's because they don't know how to do it. So you change the parameters, and we're going to talk about the gamma and everything. You have to mention these things. You cannot pretend that you have done nothing to the images. This is something you can mention at the top of the material method. All the images have been acquired without saturation and without underexposure, and the brightness and contrast has been adjusted. So it can be like a batch thing. If you write that all the images have been acquired to have no saturation, no underexposure, you send very strong signals to the reviewers that you know what you're talking about. So they will be less worried and less scrutinizing you.
Okay.
Okay. So the intensity information. So yes, the intensity information recorded in the undersaturated or saturated or underexposed areas is absolutely incorrect. The size of the saturated or underexposed objects is likely incorrect. It depends where the saturation or underexposure is. And also the number of objects may be incorrect if they fuse or if they disappear. And the correct intensity and size information cannot be recovered after acquisition because you have not acquired it, so it's not possible. So they are likely to be rejected, like those that are heavily saturated or underexposed are definitely likely to be rejected. And there is more and more demands from journals to have, because of this problem of irreproducibility, to have proper images and well documented. So do not believe your eyes. Keep the saturation and underexposure mask. on all the time and mention in the material what that the images are not saturated or underexposed. And so please set all the acquisition parameters to have no saturation and no underexposure at all. So yes, oh, it's going back. Sometime I have twice the same thing. I don't know what. And the last little bit in here. So now I'm looking at what it is. We're going to take a tiny break. So I see you guys again at, let's say, 13 past, so 15 past. Okay? If you have any questions, I'm here.